vascularis. So what is stria vascularis? Stria vascularis basically, this is the stria vascularis. It is a highly vascularized and multi-layered tissue, and it forms the part of the lateral wall of the scala media, and it plays an important role in the cochlear hemostasis. How does it do it? Basically, it maintains the homeostasis of the cochlea by generating the endocochlear potential, because it maintains a unique ion composition of the endolymph which is present in the scala media. And there are different types of cells which are present in this stria vascularis. Let's say there are marginal cells, intermediate cells, basal cells, and so on. So keep in mind that the function of the stria vascularis is to maintain the cochlear homeostasis by generating the endocochlear potential, because for the sake of the endocochlear potential it maintains a unique ion composition of the endolymph which is different from that of the perilymph present in vestibuli and tympani. So the functional units of cochlea, those were spiral ganglion, uh, this stria vascularis, and the organ of Corti. Organ of Corti, that could be considered as body's microphone. It's a neuroepithelial structure. It is the receptor organ for hearing, located on the basilar membrane in one of the three compartments of cochlea. Right. So let's have a look at the structure and the function of the organ of Corti. The organ of Corti, that consists of thousands of sensory hair cells which are attached to the basilar membrane and also to various supporting cells. Now, as far as function is concerned, the hair cells which are located in the organ of Corti, they transduce the mechanical sound vibrations into the nerve impulses, and this is how the neural signals for hearing they are generated. Right. The functional components of the organ of Corti. The functional components are the hair cells, which are inner and outer hair cells. Then there are supporting cells also, and the basilar membrane, of course, and the tectorial membrane. That is very important. So, what is this tectorial membrane? This tectorial membrane, that that forms the you can say the roof of the organ of Corti, and you see that it is in contact with the processes the stereocilia of the hair cells. It is in direct contact with these processes. So, it is in direct contact in such a way that these processes, they are stimulated by the movement of the tectorial membrane in relation to the vibrations which are produced in the endolymph. So when the vibrations, they are produced in the endolymph, which is present in the scala media, this tectorial membrane, that shows movement. So when this tectorial membrane, it shows movement, it causes the stimulation, it causes the the movement of these processes sideways. And this is how the hair cells, they are stimulated by the movement of these stereocilia, or the processes of the hair cells, that we will discuss in detail later. Okay, I tell you more about these hair cells. All right. You see, there are hair cells which are present in the inner ear, in the scala media, the in the cochlear duct of the cochlea, and there are two types of the hair cells. There are inner hair cells, and then there are outer hair cells. Sensory nerve fibers, they are distributed around the cell bodies of these hair cells, and both inner and outer hair cells, they have short afferent and long efferent nerve fibers. So they have got afferent and efferent nerve fibers. The afferent nerve fibers, they are shorter, while the efferent nerve fibers, they are long, which are carrying the signals all the way to the CNS. Now, looking at the inner hair cells, they are flask shaped in such a way that they are broader than the outer hair cells, and these inner hair cells, they are arranged in a single row, and each hair cell, it has about 100 stereocilia. uh, Among these stereocilia, one cilium that is larger, and that larger one, that is called the kinocilium. Right. The inner hair cells, they are responsible for the sound transduction. It means that they are responsible for transducing the mechanical energy into the electrical energy. So basically, these are the primary cells which cause the generation of the action potential in auditory nerve fibers. Outer hair cells. So, the structure of the outer hair cells, that is very much similar to that of the inner cells. Although the mechanism of action, that is a bit different. Now, these outer hair cells, they show electromotility. And what does that mean, that they show electromotility? It means that these outer hair cells, they shorten during depolarization, right? And they are elongated during hyperpolarization. So when they are shortened during depolarization, what happens, that they further pull the basilar membrane upward, and this is how they amplify the motion of the basilar membrane. So what happens in the electromotility of the outer hair cells? That they shorten during depolarization while they are elongated during hyperpolarization, right? Now this electromotility, that is due to the presence of a contractile protein which is called prestin. Uh, and this action of electromotility, it facilitates the movement of the basilar membrane and increases the amplitude of the basilar of the movement of the basilar membrane. And this is how they serve the function of the sharpness of sound. They are responsible for the sharpness of sound by showing this electromotility. And I told you that what is this electromotility? That they shorten during depolarization and they are elongated during hyperpolarization. Okay? So this is called the electromotility of the outer hair cells. And uh, by performing this electromotility function, these outer hair cells, they provide an extra gain of 40 to 50 decibels to the hearing system. And for the same reason, they also perform the function of the tuning of the auditory system, right? So, on the basis of the all the data given about the inner and the outer hair cells, now you have to uh, draw the uh, column of differences between the outer and the inner hair cells. You can give the difference in the shape, as the outer hair cells, they are flask shaped, but the inner hair cells, they are broader than the outer one. So you can say that the outer hair cells, they are flask shaped, and the inner hair cells, they are goblet shaped. um, but a more important difference is that only 5 to 10% of the outer hair cells, they have got the sensory afferent supply, while 90 to 95% of the inner hair cells, they are sensory afferent fibers. They have got sensory afferent fibers. um, then if you look at the applied physiology, then the outer hair cells, they are more vulnerable to trauma. And what kind of trauma? The damage or the trauma which is caused by the ototoxic drugs. Your inner hair cells, they are more resistant. It means that they are less vulnerable to the trauma caused by the ototoxic drugs, right? So you have to make a column of the differences between the outer and the inner hair cells. Keep in mind that the outer hair cells, they allow us to hear low intensities, while the inner hair cells, they respond to medium intensities and also to the high intensities. And inner hair cells, they are more passive, while the outer hair cells, they perform an active function, and they also help the inner hair cells. Then you can also give the difference in the number of the outer and inner hair cells. You see that outer hair cells, they are more in number. They are around 12,000, while the inner hair cells, they are only 3500, around 3500. And since the outer hair cells, they are more in number, so they lie in 3 to 4 rows, while the inner hair cells, they form only a single layer. So that was all about the hair cells. Now we move on to the stereocilia. What are these stereocilia? Both the inner and the outer hair cells, they contain apical stereocilia. And these stereocilia, they are fundamentally important for the hair cell transduction. Now, these stereocilia, they increase in length from the cochlear base toward the apex. The inner hair cell stereocilia, they are approximately twice as thick, and they are more, you can say they are more club shaped than the outer hair cell stereocilia. The more important thing is that there are lateral crosslinks between the stereocilia, which these links, they attach the stereocilia to one another, right? So this is how these stereocilia, they are connected to each other because of the lateral crosslinks. The outer hair cell stereocilia bundle, it forms a V-shaped, or you can say a W-shaped pattern, whose angles, they become more acute toward the cochlear apex, and they form the that's enough. Anyway, so while the inner hair cell stereocilia, they form a slightly, you can say, curved bundle. So the shape of the arrangement of stereocilia in the outer and the inner hair cells, that is also differentiating. I hope that you get a picture of the hair cells and the stereocilia in your mind. So these were the functional units of the cochlea. I told you about the organ of Corti, and very important in the organ of Corti are the hair cells. And we mentioned that there are two types of hair cells, inner hair cells and outer hair cells. And you know the differences between the inner and outer hair cells. And then there are stereocilia, which are the processes which are present on the apices of these hair cells. Then I told you about the stria vascularis and why the stria vascularis, that is important because it maintains the homeostasis of the cochlea by generating the endocochlear potential. Now, what is spiral ganglion? The neuron cell bodies which innervate the inner and the outer hair cells, they are located in the spiral ganglion. Now, the bony channel that connects the ganglion, it travel in a spiral direction toward the apex of the cochlea, and that is called the Rosenthal canal. Now, you see that approximately 90% of the spiral ganglion neurons, they are so important that they are ensheathed by around 50 lamellae of myelin, Well, your spiral ganglion, it gives off the nerve fibers which will carry the signals from your ear all the way to your CNS. Right, and then there are mm, then there are also some sporting cells in the inner ear. You see these sporting cells, and they have got different names, such as the phalangeal sporting cells. There are some outer marginal cells which are called Hansen cells. Then there are Claudius cells. Then there are border cells of Held, and so on. So, what are the rods of Corti? The supporting cells. Um, Now, these supporting cells, they are the inner and outer pillar cells. The pillars of the cells, they form series of arches, right? They form the series of arches which enclose a triangular tunnel which is called Corti's tunnel. The basilar fibers, then the rods of Corti and the reticular lamina, they form a rigid unit, and then they move as a single unit. Okay, the whole structure, this whole structure, this moves as a single unit. So whenever the basilar membrane it shows movement, this whole structure it moves as a single unit. So when this thing moves, it causes the movement, the basilar membrane it moves up and down, it will cause the movement of the stereocilia sideways. And when the stereocilia they move, they that movement result in the generation of the electrical signals. Now, once the signals are generated, how the signals they are carried to the CNS? Cochlea has got innervation. You see the cochlea that has got its nerve supply. It means that it is innervated, and the cochlea that is innervated by three types of nerve fibers. There are afferent fibers, mean the sensory fibers. Then there are efferent fibers also, which are the bringing the orders or the information from the CNS back to the ear. And then there are autonomic fibers also. The autonomic nerve fibers, they do not appear to be present within the organ of the corti, but they are associated with the blood vessels and the nerve fibers of the modulus spiral lamina, right? And there are the important ones are the afferent nerve fibers and the efferent nerve fibers. And I already told you that which type of hair cells they have got more afferent nerve fibers and which one they have got more efferent nerve fibers. Right, now we will have a look at the cochlear fluids. These cochlear fluids, these are the extracellular fluids. And these fluids are endolymph and perilymph. And they have, as you can see in the figure, that they have different chemical compositions, right? The perilymph, that is present in the scala vestibuli and the scala tympani. Both these chambers, they are filled with the perilymph. And perilymph, it is similar to the extracellular fluid in composition. And how it is similar to the extracellular fluid? Because it has a high concentration of the sodium ions, while scala media, that is filled with the endolymph, right? uh, Now endolymph, it contains high concentration of the potassium, and it has low concentration of sodium. Why it has high concentration of potassium? It is due to the continuous creation of potassium ions by the stria vascularis into the scala media. So, uh, again, you will have to draw columns of differences between the perilymph and the endolymph. Uh, you see that the perilymph, it resembles the extracellular fluid, while the endolymph, that resembles the intracellular fluid. Then the location, that is different. The perilymph, that is present in the scala tympani and scala vestibuli, while the endolymph, that is present in the scala media. And in the perilymph, the major cation, that is sodium, while in the endolymph, the major cation is potassium. Now, I give you a very important difference in perilymph and endolymph, and that is that the perilymph, it has a potential, or you can say that it has a voltage of 0 millivolt, while the voltage, or you can say the potential of the endolymph, that is around plus 80 millivolts. So there is a difference of the voltage between the endolymph and the perilymph, and the difference is around 80 millivolts. This is very important in the understanding of the endocochlear potential. Now, what is endocochlear potential? Uh, you see, this endocochlear potential, that is also called the endolymphatic potential. It is the electrical potential which is developed in the fluids outside the hair cells. Basically, what happens? That an electrical potential, a difference, an electrical potential difference, a voltage difference of plus 80 millivolts, that exists between the endolymph and the perilymph with the positivity in the scala media. Okay, I already told you that the perilymph, it has a voltage of 0 millivolt. Endolymph's voltage is around 80 millivolts. So the voltage difference, the electrical potential of 80 millivolts, that is there between the endolymph and the perilymph, right? And the difference, the positivity in this difference, that is present in the scala media. It means the voltage of the scala media, that is uh, outside the cells, that is around 80 millivolts. And why there is this difference? This difference is basically due to the continuous excretion of the potassium ions by the stria vascularis into the scala media, right? So you see that there is a difference in the inside and the outside of the cells. There is a difference of the electrical voltage between two fluids even, and that difference is around 80 millivolts. Now we will see that how the endocochlear potential is generated and what is the cause for the generation of the endocochlear potential. Now you focus on this figure. The cause for the endocochlear potential, it is the difference in the potassium concentration of perilymph and endolymph. Because your endolymph, it contains way more potassium ion concentration. The value of the endocochlear potential, that is around 140 millivolts to 150 millivolts. You see, what is the mechanism? Uh, that the lower portion of the hair cell, that is dipped, or you can say that is bathed by the perilymph. Okay, while the head portion, this head portion, that is dipped by the endolymph. And you already know that there is a difference of plus 80 millivolts between the endolymph and the perilymph. Endolymph, it has a positive potential of endolymph. This has a positive potential of plus 80 millivolts. But inside the cell, ठीक है, because of the dipping of the basis into the perilymph, now inside the hair cell, the electrical potential, that is the electrical potential which is, this is always present on the inner surfaces of the membrane. Now inside the hair cell of the organ of the Corti, this electrical potential, you can say the RMP, that is around minus 60 millivolts in comparison to that of the perilymph. So here the electrical potential is minus 60 millivolts. Outside here the electrical potential is plus 80 millivolts. So if you make it from the minus 60 to plus 80, that will give you an endocochlear potential, that is the difference in the inside and the outside of the cell, that will become around 140 millivolts, because you take this minus 60 millivolts to the zero level, that makes the 60 millivolts, and then add another plus 80, so that will become 60 plus 80, that is equal to 140 millivolts endocochlear potential. So this is how you get 140 millivolts of endocochlear potential, which is the difference in the inside and the outside of the hair cell. See what a great difference between the inside and the outside of the hair cell. Why this greater value of endocochlear potential is required? The high potential difference sensitizes the hair cells so that the excitability of these hair cells, that increases. It means that the spectrum is broadened basically. The spectrum of the sensitivity of the hair cells, that is broadened. Even if there is a very minor magnitude of the stimulus, which causes a very small change in the electrical potential between the inside and the outside of the hair cell, it will be detected. So, this is how the endocochlear potential of large value, it sensitizes the hair cells. That is just to achieve a greater excitability of the hair cells. And then also it increases the response of the hair cells, even to the slightest movement of the stereocilia. Well, I hope you understand this concept of endocochlear potential and why this endocochlear potential is important. Next. Right. So, that was your today's lecture. And if you have any questions, you can ask me directly. Next time, we will look at the mechanism of hearing, and the we will start from the events which are involved in the hearing, and then we will move on to the sound transmission. uh, Now, this is enough for now. So, okay, take care.